Hello, and welcome to the Clinical Care Options Oncology podcast. I'm your host, Ryan Topping. Today's episode features expert answers to healthcare provider questions surrounding CAR T-cell therapy for patients with hematologic malignancies. This episode is part of a larger educational program titled Current and Emerging CAR T-cell Therapies in Cancer Care. During this podcast, Dr. Caron Jacobson from the Harvard Medical School and the Dana-Farber Cancer Institute in Boston, Massachusetts, Dr. Jae Park from the Memorial Sloan Kettering Cancer Center in New York, and Dr. Noopur Raje from the Harvard Medical School and the Massachusetts General Hospital Cancer Center in Boston, Massachusetts, discuss important topics including when CAR T-cell therapy should be considered, selecting between 3 approved products for relapsed refractory large B-cell lymphoma, use of CAR T-cell therapy for multiple myeloma, and use of steroids in managing CAR T-cell therapy-related toxicities. Please visit the show notes for this episode for a link to the complete program, including downloadable slide sets, expert commentaries, and an on-demand webcast from a live CCO event. Now, let's get started and hear what the faculty have to say on these important topics.
Thanks, everyone, for the opportunity to talk to you today about CAR T-cell therapy. In your experience, at what point in treatment journey are physicians considering CAR T-cell and diffuse large B-cell lymphoma? My answer to that is it's never early enough. I mean, many patients will be referred at the time of initial relapse, which I think is fantastic, and that should be the way, but even before they start salvage chemotherapy, but sometimes patients are referred later. And because we know patients do better if they have lower tumor burden, both in terms of efficacy and toxicity, the earlier you refer these patients, the better. One question is how to decide between 3 approved CAR T-cell therapies for large cell lymphoma. And there are a number of factors that you need to consider. So one is that axi-cell has the fastest and most reliable turnaround time of the 3 agents. And so when you have a patient with rapidly growing high volume lymphoma, you may reach for that because you know that cells will come back to you in the shortest amount of time. But it also has greater toxicity than Tisacel or lisocabtagene maraleucel. And as a result, if you have more time to wait and the patient has medical comorbidities or they are older, you may reach for one of these other agents. They all have great efficacy. What qualities of bridging therapy are most preferred? I think the answer is going to be very patient dependent. It depends on the patient's comorbidities. It depends on what therapies they've seen before and what their need for bridging is. I think in general, you want to avoid things that are going to cause excessive toxicity without great efficacy. So things like steroids and radiation are great options. And then at least in lymphoma, some of the newer agents like polatuzumab, where the efficacy is very high and the toxicity is low, are also attractive agents. Is there an age limit within the geriatric population? The bottom line is very, very elderly patients can be treated successfully with CAR T-cell therapy. So whether there's an age limit will be center dependent, but I would say that age is not a contraindication for CAR T-cell therapy. And for patients who are not receptive to chemotherapy, are there lymphodepleting regimens that don't involve fludarabine and cyclophosphamide? And the answer to that is the only other regimen that I think is part of a U.S. package insert is bendamustine chemotherapy that was tested on the Juliet study of tisagenlecleucel. But fludarabine and cyclophosphamide are by far the preferred lymphodepleting regimens. Is slot availability an issue with CAR T-cell therapies? Not as much in the commercial setting. It is an issue on clinical trials for sure. What fraction of patients are ineligible for apheresis? So depending on the product, it's going to depend on the product. So for axi-cell and for liso-cell, you just need to have an absolute lymphocyte count of 100. The absolute lymphocyte count has to be a little bit higher for Tisacel. So that's usually the reason that someone would be ineligible for apheresis. Would there be a time where patients could have cells collected right after diagnosis but prior to first-line therapy and be held? We get asked this question a lot. I think it's a difficult question to answer because because 70% of patients with large cell lymphoma will be cured with upfront chemotherapy, and that's a lot of cells to collect. Plus, many patients will relapse more than a year after their completion of their therapy, and we don't know what the viability of these cells will be at that time. What are outcomes for CD19 negative relapse? What would the CAR target? So patients who have a CD19 negative relapse so I think this may get to some of the approved CD19 therapies. I'm not sure what the question is asking. But if you use a CD19 therapy and then the patient has a CD19 negative relapse, I would not recommend CAR T-cell therapy. And it's a reason I would not use CD19 directed therapies prior to CAR T-cell therapy. Second infusion of CAR T cells in large cell lymphoma hasn't really proven to be terribly effective, although in follicular lymphoma, it appears more effective. What endpoints are going to be most important for CAR T cells to replace stem cell transplant in second-line DLBCL? The primary endpoint of all of these studies is event-free survival, and in DLBCL, that's a very important endpoint because it probably reflects cure. All of these trials either allowed crossover or patients functionally crossed over to the CAR arm if they didn't respond to second-line chemotherapy. And so overall survival will be a difficult endpoint. When do CAR T-cell patients need COVID vaccination or other vaccinations? We're vaccinating all these patients 3 months after CAR, but I'm not sure that they're responding. We're testing this now. And so I suspect we're going to need to give all of these patients boosters at some point. All other vaccines we're generally reserving for 1 year after CAR T-cell infusion.
What role will consolidation post-CAR T-cell in multiple myeloma play? What regimens do you anticipate will be useful in consolidation? I think we are doing a bunch of sort of umbrella trials right now to explore this question, and whether we use something like a BITE strategy for a stipulated duration of time is something time will tell. But you're absolutely right. This is an area of active interest so that we can try and maintain the duration of response. What I would really like to see, we have allogeneic CAR T cells in the myeloma space as well. And one of the challenges of an allogeneic product is persistence of CARs or persistence of the T cells there. So one way of thinking about this is start with an allo product, go in later on with an auto product or vice versa, or use an auto product followed by a bispecific T-cell engager. So all of these are possibilities which we are exploring. What are the most common reasons for ide-cel to fail quickly after infusion and progression after 2 months? I think the biggest challenge there is, we've used CAR T cells sort of after you've seen lots of different treatments already. We talked a lot about T-cell fitness, and I do think T-cell fitness is extremely important. We have data now to show that the less heavily pre-treated you are, the fitter you are. There was a question asked of Dr. Jacobson, should we be collecting CAR T cells early? And, logistically, that's really tough to do. But I do think the less exposed patients are to some of these treatments, the fitter those T cells are. And as we show that these drug products are really useful in the relapsed refractory state, we are already moving them on into earlier stages wherein we're going to have fitter T cells. Is there data for cilta-cel working after ide-cel has failed? Again, a good question. We don't have data because cilta-cel is not yet approved. But, given that you're seeing much higher binding, and given that you're seeing cilta-cel targeting two different epitopes what I can imagine seeing is some, you may be able to see more efficacy with one product after another. Whether we will be able to do it on all patients, it's unclear at this moment, and whether we'd be able to sequence them or not sure as yet.
Do we have a different toxicity management for different products and then the different patient population? That's actually a very good question. We are actually dealing with this as we speak. We want to have a somewhat of a uniformed IEC, immune effector cell management toxicity, but as we have seen that not all products have a similar or the same time frame of a toxicity profiles, and then it's actually even bigger issue with the TILs and TCRs where the management has been slightly different. Steroid we are less likely to use. So I think right now for the investigational therapies they have their own, but the commercials and then the post-approval, we do have some of a loosely uniform criteria to use, similar to the form that we just discussed about grade 2. And there are things that we definitely do agree on. But sometimes we may intervene for grade 1 CRS and grade 1 neurotoxicity if we know the product is associated with a high risk of a severe neurotoxicity, if that's a pattern. And if the patient is also a high-risk patient, meaning high LDH, high tumor burden patients, or older patients that we are worried that if they were to develop high degree of a toxicity, their recovery may be delayed. So there are some wiggle room, and they have to be. So I think these are the factors to consider. But because of that, it's difficult to have a one uniform policy for everyone. I think another question do have one is EEG appropriate? I do think we do them often for encephalopathic patients, so typically getting a grade 2 or C, grade 3 ICANS. The patients, I think neuroconsult and or engaged neuro team that we have found to be very helpful for them to intervene and follow these patients. We also tend to do baseline neuro exams because we also, baseline exams is helpful to see if they do develop toxicity, whether these are new or they're pre-existing. So I think these are also kind of helpful things. But EEGs we do routine, although we really haven't found much of a seizure during this. More of a diffuse slowing is the most common finding that we do see. Yufei asked, can you comment on the ICANS-induced patient death in a recent prostate cancer clinical trial? What do you think is the future toxicity with solid tumors? The good question. I don't think that much details really kind of came about those, and it's a different target. I think it might have been the PSMA and a different construct there too. I think there are more toxicity do come about. ICANS has not been as common in the solid tumors. It's more common in adult ALL, CD19, but it has been seen in solid tumor studies and CAR studies as well. So I don't think that enough of the details to know whether there was a true neurotoxicity or the HLH or the macrophage activation syndrome, and then the progression of those, and then having encephalopathic pictures from the liver failure. So those are always very difficult to distinguish. So it's a little hard for me to comment on. Standard antimicrobial prophylaxis. We do use them because these patients tend to get a neutropenic from their conditioning chemotherapy from Flu/Cy. So we do have a standard antiviral prophylaxis, fungal prophylaxis, and the neutropenic if they're outpatient and certainly neutropenic. Fungal prophylaxis has varied by disease service, lymphoma versus leukemia, and then the transplant team, but they do tend to get some type of fungal while they are neutropenic. Steroid can reduce the efficacy of CAR T. How do you manage CR as well, considering this faster? Let me see. So the CAR T, the steroid use, I think that's one has been a concern several years ago when we were used to do this, the big early stages of a therapy where early use of a steroid can reduce the CAR T cell efficacy. I think we have now more data to suggest that these, at least also with tocilizumab too, do not really seem to impact initial response rate. I think duration of remission is something that is also, we're getting emerging data, more data showing that it also doesn't seem to be impacting those. So we are less worried about using steroid. But whenever we use them, I like to use them as short as possible. But what I also found is if you get there too late, grade 3 neurotoxicity, you end up using steroid even longer. So I think getting there a little earlier, you can actually get away with a shorter duration, shorter cumulative doses of a steroid. So I think that's where the field is moving, and steroid is just only one of them. There are other strategies to prevent the CRS and neurotoxicity. The question is, if you see ferritin over 10,000, does it qualify for HLH? I don't think so, not always. I think you have to put this in a context, and Caron and Noopur may have other thoughts on this one too. I think not all 10,000 ferritin is the high, but we also have seen it in the very severe CRS and neurotoxicities too, and not necessarily HLH. I think HLH is these patients are clinically also very sick too. We usually accompanied by high fevers. And ferritin 10,000, when you see the fever resolutions and some neurotoxicity, I don't think those are truly had 100. So it says 100,000, Leslie said, and not 10,000. But I mean, those are very high, so I'll be worried about them. I mean, it could be HLH or macrophage inactivation syndrome. Could certainly be a consideration, but I think you have to put that in a context. But I do agree that it's a very high levels, and those are the patients that will be using very high dose of how you measure if the patient is also clinically unwell, which tend to be the case. And then one person asked about the prophylaxis guideline. Can we share them? I think I would be happy to if you kind of reach out to us, any one of us. But also there was a blood publication about how I treat, I think, infectious or how I treat series in the blood, I think 2020 from Seattle and MSK from infectious disease specialists, kind of outlining some of the vaccination guidelines and then infectious prophylaxis guidelines. So that may be a valuable resource. And I think it was in 2020 blood.
I think the only other question was about BTK inhibitors and T cell fitness. And I mean, most of the data we have comes out of the CLL population where the disease itself interacts with T cell fitness. And so the effect of it's hard to dissect out the effect of BTK inhibitors on T cell fitness versus the BTK inhibitors' effect on the disease, which may actually improve T cell fitness, although people are exploring BTK inhibitors in other diseases before leukapheresis, and Noopur mentioned BB21217, which uses a PI3 kinase inhibitor during manufacturing to try to improve T cell fitness. But hopefully this will be moot as CAR T cells are approved in earlier lines of therapy.
I know. I think there's one more question, actually. I think they asked ongoing trials and evidence for using CAR T cell therapy in solid tumors. And there are many. I think the prostate cancer trial, the one was referring to as being one of them, but there are many targeting mesothelin CARs. There are EGFR targeting CARs. I think, and there are many TCRs and the TILs as too in solid tumors too. So I think we don't have as much of a data in solid tumors, but there are several that's ongoing. It's been a little bit, I think, challenging because it's hard to find a universal target that is tumor specific and has done, sparing the normal tissues. It has been less easy, I think, solid tumors. Thank you for attending.
Thank you very much, Dr. Jacobson, Dr. Park, and Dr. Raje, and thanks to you, the listeners, for joining us. As a reminder, to view the full program, Current and Emerging CAR T-Cell Therapies in Cancer Care, and to download slide sets associated with this discussion from the Clinical Care Options website, please click on the links in the show notes. As always, thanks for listening.